Hello, hello. Warning, I'm having sneeze attacks today. I don't know what that's about. So this is for week 4. The lecture link will work as soon as this is over, and the hours it takes to transfer it to YouTube will happen. Um, these extra links here I've given you just, to increase your enjoyment of the course, I hope. And I don't think any of this is testable, so we'll be sticking with the book. We are finally into neuroscience, which is my thing, and this is a good chapter. It's nowhere near as detailed as I typically like to do, but this is a lower level course, and what the hey. These are good objectives, and some of which could be turned into here we go. Testing questions. And we'll see I have a link to the concept of homeostasis because of someone who shares my name, but we are not related. Um, there's an interesting picture. Later in the book, it talks about ways to image the brain, and that's an MRI, magnetic resonance imaging, that's been colorized, which means adding color to the black and white. Um, MRIs can be quite detailed. Um, I used to get a lot of them. I forget. Oh no, that's because you remember Descartes and the pineal gland. I had a have a cyst on my pineal gland, and they used to like to image that. So here's homeostasis, um, and the phrase that should come to mind when you see this is wisdom of the body, and there's a few examples here of homeostatic systems. And by the way, some tolerance to drugs may occur for reasons related to homeostasis. And I've given you a link. Walter Buchanan, no relation. Wisdom of the Body, this is the wiki page, and there's Walter. I study faces. He's got very big gap between the bottom of his nose and the top of his lips, which I tend to think is related to being smarter. It says that the term homeostasis derived from Claude Bernard, uh, but he wrote a book, The Wisdom of the Body, and that's a phrase that comes to mind every time I think about homeostasis. The body largely wants to be left alone. Stable temperature, relatively stable glucose, relatively stable sodium levels, lots of things. Um, and again, this can relate to drug and drug addiction. You may love a drug, but the body wants to be left alone and can compensate for the drug if you let it, leading to tolerance. So that's good. And ADH, antidiuretic hormone, nice discussion. Alcohol, which, by the way, I would vote increases aggression. Uh, some of you don't agree, but just because many people even can use it without becoming aggressive, it does tend to increase aggression. All right, so components of the nervous system. We obsess over neurons, and it's got one big membrane, and the membrane uh, keeps a little ocean inside the cell, and there's a bigger ocean outside the cell, and it's one big membrane. But there are channels in it, and those channels uh, allow for magic. There are dendrites. My daughter was taking a class in dendrology because they look like tree branches. Try to appreciate, by the way, that this is three-dimensional. This looks like it's been gone over by a steamroller and it's flattened. It's three-dimensional, and dendrites can be quite extensive. And oh, geez, are they alive? They can change in seconds, which is one of the ways we adapt to the nervous system. And you have the cell body, and the nucleus is in the cell body, and that provides us with some magic. Notice that the neuron is colored orange, uh, and the myelin sheath, segments of myelin there, pieces of myelin, um, that's added on. It's not part of the axon, not part of the axon. It's wrapped around the axon. Um, and action potential will be part of this chapter. And nodes of Ranvier, back when French people did important science, those are the gaps between the segments of myelin, and they are important. And the axon terminals can look a bit like dendrites. And that's where the stereotypical synapses are. And that will be discussed later. I have something to, oh, glia, we'll do glia now. Glia derives from the term glue. And at one time, we didn't think glia were very important to the functioning of the brain. It just kind of glued the brain together, which is pretty important. But as time goes on, we are learning more and more about glia, and they do far more than simply glue things uh, together. And it says the uh, there, if you're going to get a cancer in your brain, or even a tumor, it usually derives from glia. These are dividing all the time in the brain. Neurons tend not to, although some do. Which means that if you're going to lose control over replication of cells in the brain, it will probably be the glia. And this can be a very aggressive form of cancer. The former president, Jimmy Carter, had this type of cancer, and they virtually magically cured him with a relatively new treatment for the cancer he had. And fingers crossed, he's been cancer free for a few years. And there's myelin, which I said is not part of the axon. It's on the axon. And there's an autoimmune disease, which means your immune system attacks you, and you love it when your immune system attacks foreign bodies, enemies, viruses, bacteria. But if it decides that you're the problem, it wakes up one day and says, "What's this myelin stuff? What is this?" And it decides it's a foreign body and it starts attacking it. You lose your myelin, and your neurons, your action potential moving down your axon doesn't just go slower; it can stop. And I think I have a link relevant to that, not MS, but relevant to something I had. Guillain-Barré. I had this when I was a little younger than you guys. It was memorable. Um, when I had it, I had sensory issues, I had motor issues. I couldn't open my mouth wide enough to put a sandwich in. I used to have to sit in high school and pound on my sandwiches to flatten them. I was too stupid to be afraid. It's usually self-limiting. Normally you don't die, and it just goes away. This is like a peripheral version of MS, uh, and it came and it went. It was memorable. So here's an image of Guillain-Barré, and there's a disturbing picture on the right. Your axon, which is the tan thing there, and then the myelin. You can see it. The myelin is just being ripped to shreds by your immune system. So with MS, your immune system in your central nervous system goes after your myelin. With myasthenia gravis, your immune system attacks your peripheral nervous system. Yeah, and 60, 80%. Well, I never lost my ability to walk, but if I fell down, it's like being old. I couldn't get up. I had to crawl to something and drag myself up it. Um, but it didn't hurt. I kind of thought it was funny. I was stupid. Um, okay, so that's a discussion of myelin. And by the way, the myelin in the central nervous system, which we'll come to later, is made by different kinds of cells than the myelin that's made in the peripheral nervous system, probably explaining why MS and Guillain-Barré independently attack central versus peripheral. Um, okay, blood-brain barrier. This is so important. And oops, I put in one too many dashes in my link. Um, this is something that if you play a sport that involves getting your head hit with any regularity, like soccer, your blood-brain barrier is threatened, and it's not meant to be. You're not supposed to be, whacking things off your head with any regularity. We didn't evolve to do that. And unfiltered blood is toxic to the brain, and you don't want that to happen. The blood-brain barrier is semi-permeable, largely, and by the way, the cell membrane of a neuron is semi-permeable. Some stuff is supposed to get in there, some isn't. One of the tricks to treating brain cancers is how to get drugs into the brain, because if you want the drug to attack the cancer, it has to get in there, and the barrier can be a barrier. Um, the what do I have? Oh, I have I used to, as we'll see, produce lesions and put cannulae and electrodes in rat brains, and that's an insult to the brain, and it disrupts the blood-brain barrier, and stuff leaks into the brain that's toxic and can, it can kill, but it also messes things up until the barrier reestablishes itself. And an odd but true way of looking at the blood-brain barrier is ink. If you inject ink into the blood supply of a mammal like a rat, the ink will affect peripheral tissues, and they will turn whatever color the ink is, usually blue. But the brain will be spared. It will not turn blue because of the blood-brain barrier. Um, However, if you have lesioned the brain, here's a picture. So on the bottom, there's no staining there. You're just looking at a cut of the brain, and they have done something to what presumably is the left side of the brain there, and all that blue is ink that has leaked through the blood-brain barrier from that insult, and that will last for probably about 2 weeks after the insult. And again, I've done research on soccer and cognitive testing of soccer players, and uh, it doesn't do that. It's subtle, but it's related to that. So things are leaking into the brain that shouldn't leak into the brain, and that can certainly mess the brain up and even kill parts of it. So we know boxing is bad, and we know football is bad. We've been slow to recognize that soccer is probably bad. And what delayed our understanding of that was a lot of the early research, or some of the early research, was done in Europe, where they call soccer football. And I used to see studies that say, football related brain injury. And I thought, well, yeah, football. but no, not football, soccer. Um, Not a fan. So there's neurotransmission and action potentials and channels. It's a nice discussion. It's a light discussion. Ions have charges, and when charges move across the membrane, they produce electrical fields, and you have a resting potential. Most cells are around minus 60, minus 70, just sitting there, nothing special. And then you get often, usually synaptic transmission, although it could be, if this is a sensory neuron, it could be a touch. And there's sodium channels opening. Sodium channels are very important. They are positive ions. There's more outside than in. They start coming in. And as a positive ion goes into a cell membrane, it becomes less negative. And finally, you get to the threshold. This is like lighting a fuse. And once the fuse is lit, bam, the sodium channels open, and there's a flood of sodium, which causes what I call the rising phase of the action potential. And yeah, it goes up to about plus 50 millivolts. Does this say millivolts? Yes, millivolts, MV. And then the sodium channels close, and then potassium, which is not happy with the inside of the cell being positive, because potassium is positive, and potassium starts rushing out of the cell and it's a positive ion lead. So the rising phase, the active potential is sodium. The falling phase is potassium. And as a positive ion leaves a cell membrane, it becomes more negative. And normally I would spend an obscene amount of time on the undershoot phase, but for this class, we will just skip it. If you want to get a hint as to what I do uh, when I teach my behavioral neuroscience course, uh, these are links relative to quizzes, and this is the figure I would have asked you to learn about action potentials. You can see the shape is pretty much the same. And there's sodium in on the left and potassium out on the right, and there's additional details that we're not worrying about. And this hyperpolarization overshoot that the book referred to is the relative refractory period, and I am obsessed with that. Uh, you're lucky you don't have to deal with that. Neuroscience majors became so ingrained in this, they use this symbol for a bar crawl. Um, oops. Yes. Okay, so that's a good figure. It's not wrong. It's just thin, and it talks about blocking sodium channels, and you've had this with lidocaine if you've had lidocaine, which is related to cocaine. And I just had this when I went to Hopkins, and I am so happy it works. I can't imagine how surgical medicine was done back in the day before they had local or general anesthetics. It had to be a nightmare. Physicians would amputate using saws as fast as they could go, and occasionally they would cleave off the fingers of themselves or their assistants because they were working so fast to get the surgery done in a fully conscious patient. Be very glad that you live in an era with local and general anesthetics. So if you can't open your sodium channels, you don't get the action potential. For example. Okay, there's that page. This is a really good chapter. So you have the nervous system, somatic and autonomic. Um, Guillain-Barré affected the somatic, and that tends to use acetylcholine as the neurotransmitter to talk to your striated muscles. Interestingly enough, cigarettes, nicotine, Uh, the receptors on your muscles respond to acetylcholine and nicotine. Um, Insects use acetylcholine too, and one of the ways that we naturally attack insects is to take tobacco, put it in water, like make a tea, and then put it on the critters, and they'll die. Humans, not so much. But it does affect parts of the brain that are related to addiction, which will come up later in the class. Autonomic nervous system, sympathetic and parasympathetic, and these are often related to side effects of drugs. The heart rate, vagus nerve, my heart rate is up. I don't know what the heck is going on there. Um, and there's yin and yang here, competing things. Um, so there's heart rate, sympathetic increase, heart rate with the sympathetic nervous system, and parasympathetic decreases. And heart rate was important to the discovery of chemical neurotransmission. At one time, the field believed that neurons talk to other cells electrically, not with chemicals. And that did make sense. It was parsimonious to believe that. It was the simplest possible explanation. But this guy, Otto Levy, the W should be a V. I've always said it incorrectly, and now I realize I'm wrong. I've been saying Lowy, but that should be a V because he was German. And he only won the Nobel Prize in part for this. And he could tie a bow tie. And he dreamt of an experiment he could do in the lab to test whether uh, chemicals were used in neural transmission. And he shared the Nobel Prize with somebody named Dale. And there's a picture of them in here. Oh, there they are. Aren't they cute? Dale was much bigger. Nice hats. And there's the study, and you're stimulating the vagus, and the heart rate slows, and then you take liquid that surrounds that heart. Uh, this is all been surgically removed from the frog, and you take liquid from the heart on the left, and you put it on the heart of the right, and Nobel Prize time. The second heart, which was not influenced by vagal stimulation, it slows down too. Really quite acute study. Apparently, he had to do this twice. The first time, he dreamt the solution and then forgot it, which is often the case with dreams. The second time, he made sure he remembered it. And yeah, Nobel Prize. Bowler hat. So take a look at those tables, um, and this should remind you of the yin yang idea of balance between competing forces. And in relation, that is what goes on here. For example, your pupils. Your pupils are not completely dilated. They are not completely constricted. They are a balance between those two systems. By the way, if you stopped while driving, the police will be very interested in your pupils as an indicator of whether you've been taking uh, recreational drugs, some of which will affect that. Central nervous system, largely the brain and spinal cord. There are exceptions. By the way, when you say brain and spinal cord, do not think skull and spinal column. The column is the bones. Um, there's a bunch of nervous system leading to and coming from the spinal cord that can be inside those bones. But as soon as a neuron transitions between outside the spinal cord and the inside, it it changes from being peripheral to central or central to peripheral. And by the way, the myelin that I mentioned that's on the axons, those cells change too. So you have a fair amount of peripheral nervous system in your spinal column. Uh, and what are we doing here? Oh, yes. Um, and then we have neurotransmitters. And of course, these are incredibly important to the class because drugs that are psychoactive usually interact with these folks. Um, Neuropeptides, that was my stick. I was doing with pain and pain inhibition, and neuropeptides are important to me. And there's dopamine, which is way important to this class. And there are two nuclear groups that you should know by heart: nucleus accumbens, which is part of the midbrain, and the VTA, ventral tegmental area, which is part of the forebrain. And virtually any drug that is fun and or addictive is going to affect these two areas. And we've had at least two fairly famous graduates of the university in my lab who worked on these areas. And one of them is an administrator now, but the other one continues to work on this. So VTA, nucleus accumbens, know it. And that's the mesolimbic dopamine pathway. And it's thought to be important uh, in many things. What else we got? So there's the nucleus accumbens in the forebrain, and there's the VTA in the midbrain. They're close, but that's the midbrain, which I spent most of my life in. You're also going to going to learn about the substantia nigra and parts of the striatum and frontal cortex. You're seeing a lot of that. When you think of substantia nigra, you should think, why is it called substantia nigra? Because when you slice a fresh brain open through that area, you see a dark or black area. And the first time I saw that, I was just blown away. Geez, it's black. Um, and that's death of those neurons is involved in Parkinson's disease. And as they die, the pigment in them dies. So it becomes less nigra. Lots of talking, lots of talking. Nigrostriatal, so there's Parkinson's. L-DOPA is important, so now I think we jump to what I provided you with. So there's LOE, substantia nigra, and this is just the brain is perfused. This is a cross section through largely the midbrain, and we're going to see later this area up here is where I did most of my research. And on the left is somebody who is unfortunate enough to have Parkinson's or have had Parkinson's. Of course, both of these people are dead. And compare that brain with the brain on the right, they're also dead. And you can see that the person on the right's substantia nigra is much more pigmented because it's the neurons are still there. So this is a progressive disease that we still can't figure out how to effectively deal with. And it can be, it can kill you. And it can be very debilitating. And I believe I have a video for you. It's not high quality video, but it's dramatic. Arms aren't swinging. Jeez. Being locked into that body must be horrible. Boy, this bothers me. Look at that. He's happy he's walking. L-Dopa can be a miracle drug, but unfortunately, it doesn't cure the disease. It's symptomatic treatment, and after a while, unfortunately, it stops working. I don't think I'll. Maybe I will. Uh, I will give you a link. But there was a famous movie and book. I I really recommend this book, and I had a natural attachment to Oliver Sacks. Um, I own some of his books. He was a brilliant writer, and Robin Williams played Oliver Sacks in this movie, and Oliver was quirky, and you can read about it. It's and I'm not testing on this, but there were all these people after largely the flu epidemic who had encephalitis lethargica, which you just became locked in. They didn't do anything. They were housed in uh, state institutions often, and Oliver Sacks worked there. And being a creative, probably attention deficit disordered person, he started reading about L-Dopa and knew Parkinson's. He was a physician, and he said, "Geez, L-Dopa helps Parkinson's. Will it help these people?" And the book and movie ensued. Unfortunately, it largely did not have a happy ending because the L-Dopa therapy here too uh, did not cure these patients. It gave them symptomatic relief, and after decades, they basically came back to life, and then it stopped working, and they went back to being what they were. So I'll add this to the link list for you. Just give me a second. I definitely recommend that movie. And anything else that Oliver Sacks did, he died just a few years ago. And I will publish it immediately so I don't forget. Okay, and now we're back over here, and it should be published. I'm going to recommend some other books to you. So a student wrote me and thoughtfully provided me with links um, to some relevant material, and I then buried them in links to books and movies and stuff. and I'll share that with you later. And I might sneeze. There we go. Here's the wiki entry for L-DOPA, which has the structure, which is not overly relevant to you, but you can see it's it's a fairly simple molecule, um, and it's a nice discussion. And there's medical use and uh, just take a look. Again, I hope that I'm sharing excitement by sharing these links. I'm not trying to burden you. I'm trying to convince you that there's a lot of stuff out there that could be quite interesting. And now, whoops, fairly quickly, we're going to move to a related neurotransmitter. We've already dealt with acetylcholine a bit, and there's where it is in the central nervous system, and it's discussed a lot in relation to Alzheimer's disease. But Alzheimer's is bigger than just acetylcholine, and there's norepinephrine. Now, this book uses a spelling of ceruleus that's different than what I use. Um, there's there are different spellings. Norepinephrine is a companion neurotransmitter to dopamine, and to prove that to you, uh, oops, no. Oh, by the way, if you're in my other course, these are the drugs you have to learn. You'll learn a lot of these, not relevant to you. Where the heck is it? Oh, wait. Um, well, anyway, I have the synthetic pathways for neurotransmitters in here somewhere. Uh, it's a much busier course than what you're dealing with, and it shows the synthetic pathway for norepinephrine and dopamine. You got to make one to make the other, which is why drugs that affect one also tend to affect the other. Now, we know that substantia nigra was called substantia nigra because when you sliced open the brain, um, they would scream, "Oh, look, there's black stuff in there." Well, norepinephrine is a related neurotransmitter, and it's chemically related. It's just one step away, um, and it also has color, but the color is not black; it's blue, and thus the term locus ceruleus. So we will go over here, uh, and this is upside down. I couldn't change it. But these are two adjoining sections in a human. The cerebellum would be up here, which we're not discussing. And this is the pons down here, and there are these blue areas. Now I've sliced open a few brains, and I have seen substantia nigra being dark. I have never sliced open a brain and seen the locus ceruleus, so that's a little more subtle. But it's interesting that these two neurotransmitters that are synthetically related to each other, both color the brain, and they're interesting. They're interesting. And by the way, if you're looking for the origin of the term locus ceruleus, it's blue. So here's the wiki page on cerulean blue. just in case you forgot what blue looks like. And we're going to get to that in a second. Yeah, right here. Serotonin. Serotonin is in the news a lot in relation to depression and sleep and pain and a bunch of things. And this is what I used to do research on. There's Prozac for treatment of depression. Um, the I guess since I'm touching it, the success of selective serotonin reuptake inhibitors, SSRIs, which should really have been there, um, that says how it works. So they are drugs that selectively keep you from turning off serotonin by taking it back into the neuron. And so you speak with your own serotonin, but you speak louder and longer. Um, and very popular drug. And other drugs, also, at least in part, work by dealing with serotonin reuptake. Now, the part of the brain that's relevant, the parts of the brain that are relevant to serotonin are the raphe nuclei. Notice that's plural. Raphe means seam, and you only have one seam. Somewhere you can probably see parts of your body where your hair, goes to the left of your seam and to the right of your seam. But and the raphe nuclei, you have a left and right substantia nigra. You have a left and right arcuate nucleus, left and right everything except the pineal gland, which is why Descartes put your soul there. Uh, but your raphe nuclei, there are plenty, but they're all on the midline. And I did research on those. And there's one figure that's relevant. There's a cross section of a rat brain, which might look vaguely familiar in terms of both, well, certainly locus coeruleus ballpark, and even substantia nigra. This is sort of between those two. And PAG means peri, which means around, aqueductal gray. So it's the around the aqueduct gray, and that little hole in the middle, that's the aqueduct. And DR is dorsal raphe. So I was doing pain inhibition by sticking electrodes straight down the midline here. And then naloxone comes up in this chapter quite a bit, an important drug for saving people's lives from opiate overdoses. And the white bars are what naloxone does if the electrodes are up here in what I call the dorsal area, because it is dorsal. It means, back or fin, dorsal fin. And the dorsal raphe, unfortunately, is in the ventral area. And when the electrodes are down there, you can see that as you increase the dose of naloxone, that does something. So that bottom area that includes the dorsal raphe nucleus, which has serotonin in it, that's important for pain inhibition that seems to involve opioids and can be blocked by naloxone or weakened by naloxone. So the higher the black bar goes up, the more weakening there has been. And now here's another raphe. So this is the raphe magnus. Actually, it's not there because I destroyed it. But you might guess that the raphe magnus is called magnus because it's big. And the dorsal raphe and its area talks to the raphe magnus and related areas. And this is how pain inhibition is transferred to the spinal cord. So it's from the dorsal raphe to this area that includes the raphe magnus. And I blew that up. And as I blew up more and more of it, percentage destroyed, raphe magnus, the dots go up, meaning you need to work harder and harder to put analgesia, deal with analgesia. So Raphe Magnus and Dorsal both contain serotonin, and the Raphe Magnus area is the primary area that sends axons to the spinal cord and can inhibit pain. So this stuff is important to me. Um, and then there's a bunch of other neurotransmitters. GABA is huge. Alcohol affects GABA. Um, and GHB, which can be used as a date rape drug. It is used in Europe for birth and babies. If used medically, it's quite safe. It's pretty much safe. And then so glutamate, GABA is an inhibitory neurotransmitter. Glutamate is an excitatory neurotransmitter, and glutamate is thought to be, well, it's important for a lot of things. And notice that the nucleus accumbens comes up here. More neurotransmitters. Um, the book uses the term endorphin or endorphins. I prefer the term opioids. And they're definitely important for pain relief and a bunch of other things. And oxytocin has been referred to as the hug or love hormone. Something that hugging doesn't happen much during COVID outbreaks. And MDMA. Yeah. So these are relevant. And endocannabinoids, endogenous marijuana type drugs. These are very important. Remember my figure dealing with brain stimulation and this upper area here, which is the white bars over here. Naloxone doesn't affect that. So whatever, you're getting pain relief from up there, but naloxone doesn't touch it. So we called it non-opioid. That's endocannabinoid. And we found that decades ago. But the federal government, as you know, is not encouraging people to study cannabinoids, and their marijuana is Schedule 1. But stimulating that dorsal area also goes to the spinal cord using a different pathway, and it inhibits pain. And the ventral area, the opioid area, you get tolerance and cross tolerance. The upper area, non-opioid, because we didn't know what it was, but we now know it's largely endocannabinoid. You don't get tolerance or cross tolerance. I was nuts for that area, but the rest of the world was obsessed with opioids. So we didn't study that very much. And that's too bad. But they're studying it now. All right, so there's a partial life story of neurotransmitters often stored in vesicles. Remember, the cell membrane separates an ocean outside and an ocean inside. So those vesicles have to fuse with the cell membrane and release their contents. The synaptic cleft, that gap, is way, way smaller than is shown in the picture. But neurotransmitter molecules have to get across there and hit receptor sites. So drugs can work in many, many ways here. You can affect synthesis, you can affect storage in the vesicles, you can affect release from the vesicles. You can directly affect the receptors, either stimulating them or blocking them. And what they don't show in this picture is that little neurotransmitters sometimes are reuptaken into the axon terminal, reuptake. And, if you're following this, you heard me say, or you saw the book say, serotonin specific reuptake inhibitors. So in that case, the little orangey balls would be serotonin, and there would be active transport mechanisms that would look for the serotonin and drag it back inside the presynaptic terminal, or the neuron that released it, and of course that deactivates it in the sense that the receptors can't see it anymore, but you get to reuse it, which is kind of cute. So you don't have to make replacements. Other neurotransmitters are deactivated by enzymes in that gap, and they're destroyed there, which is fast, but also expensive because you have to make your neurotransmitter again. But if you want things to happen quickly, that's probably how you turn things off. Okay, so now we have more parts of the brain, and basal ganglia is important for motor activity and lots of other things. So, um, cerebral cortex, medial forebrain bundle in that picture is imaginary. Um, it's an important pathway for reward, and so that's why it's there. The midbrain is the midbrain. It's between the forebrain and the hindbrain. So the forebrain is all this mess up here, and the hindbrain is this mess down here. Um, and there's the cerebellum, which I did mention, but the book doesn't talk about very much. The hypothalamus. The hypothalamus is called the hypothalamus because it's hypo to the thalamus. So if you are hypothermic, your temperature is low, and you are below the thalamus. Pons is behind the midbrain, and your locus coeruleus is in there. The reticular activating system actually runs up through most of that midline area, not just in the pons. And medulla, we often just say medulla, but it is more formally the medulla oblongata. There's a discussion of the limbic system, which is important to a bunch of stuff relevant to this course. Uh, Emotion, memory, for location, physical activity. Uh, it mentions two parts of it, the amygdala, or the amygdaloid complex, as I like to say, and the hippocampus. The amygdala is in your temporal lobe. It's pretty lateral. The hippocampus kind of it hippocampus is related to the structure of a seahorse. It starts in your temporal lobes but goes more medially. It's very important for memory. And there's our friend, the midbrain, uh, and the brainstem. We like the brainstem. Area postrema. This is what you vomit with. So you might think about what you would predict the blood-brain barrier would be like at the area postrema. So if you've got cells in there that are supposed to detect toxins so you can vomit them up, would you like a weak blood-brain barrier there or a strong one? And of course, if you had a strong one, you would never detect things because they wouldn't get in there. So yeah, you feel like you're vomiting with your stomach, and mechanically you are. But the area postrema is the part of your brain that did it to you. So when you overconsume alcohol, for example, that's your area postrema trying to get rid of toxins. Now we're talking about synthesis of neurotransmitters, uptake, which is not reuptake, where you uptake molecules that are relevant to the synthesis of neurotransmitter. Oh, look, I didn't have to find it in my other class. So there's synthesis of both dopamine and norepinephrine. Tyrosine to dopa, which is of course related to L-dopa, dopa to dopamine, and then dopamine to norepinephrine. So that's an important pathway. And look at the structures of dopamine and norepinephrine. Don't they look similar? Of course, they almost have to be because they're only one synthetic step different from each other. Dietary tyrosine is part of this package. And with Parkinson's disease, you might ask yourself, well, why don't we just inject people with dopamine? It doesn't do a good job of getting into the brain, blood-brain barrier, but L-dopa does. So there's a discussion of enzymes and how enzymes can facilitate the synthesis or degradation of a neurotransmitter. They're important, and there's a discussion of the synapse. And here's a nightmare that in my regular behavior neuroscience class you would have to confront, not so much here, but there are receptor types. So serotonin, geez, I think there's over 12. And there are different GABA receptors, and GABA is important to a number of things. And different receptors do different things. 11 nicotinic receptors. Jeez. Neuroscience will not run out of things to do anytime soon. So there's a neurotransmitter being destroyed by an enzyme. That's largely how we get rid of acetylcholine. Quick action on your muscles and quick action off. If you couldn't turn it off, your muscles would contract and not relax, and that would kill you. Agonist and antagonist. Now, the book emphasizes receptor effects. So if a drug goes into a neurotransmitter receptor, like an opioid receptor, and acts like an opioid or an endorphin, same thing, then that would be an agonist. But if a drug goes into that receptor and just sits there, it's like somebody put a key in your lock, but the key doesn't do anything. It's the wrong key. But while that key is in your receptor, you can't put your key in. And that will antagonize your ability to unlock your door. And that's naloxone. And there it is right there. Opioid antagonist naloxone. So opioid, again, is what I would prefer you call your own endogenous body-made chemicals, and drugs that are taken from outside. I would prefer the term opiate, and naloxone works on both because it works on the receptor site. And I've done a lot of research with naloxone. So then, there's sort of a philosophical discussion that starts with humors. And can you guess why monoamines are called monoamines? Because they have one amine group. And norepinephrine and dopamine are monoamines, and they're important to lots of things: depression, schizophrenia, elements of Alzheimer's, although acetylcholine is not a monoamine. Right. Things were simpler in my day. I think you could count neurotransmitters on one hand, maybe a bit of two, and now we have like over 100. And then you add the complexity that we know there are multiple receptors. It's complicated in there. Billions of neurons, more than 100 neurotransmitters, many of them having multiple receptors. It's complicated. Brain imaging. Um, there's some social justice there you can look at. I'm not going to worry about that too much. Although I could test you on this, it's just that the book does a good job. And it talks about MRIs and PET scans and fMRIs. MRIs, I think when early in my life, we had CAT scans, which use X-rays, and they are still used in hospitals. They can image some things pretty well. They have low resolution MRIs, magnetic resonance imaging. These are the things with the huge magnets. I remember I went into an MRI and forgot to take my, at that time, wedding ring off. You go in there with something that can be affected by a magnet, and your life can be in jeopardy. Fortunately, my wedding ring was as pure gold as it was supposed to be, and it just sort of tingled. But there are pictures of those big buffing machines they used to polish and clean floors. The wandering into an MRI suite with the MRI on, and that giant machine getting slammed into the hole. Firefighters going in with breathing apparatus in that are magnetic and getting the breathing apparatus with the firefighter attached being slammed into the MRI. Those are serious magnets, and they are improving these MRIs, as we speak. And to improve them, they generally make the magnets stronger. I was almost involved in PET scan research, positron emission tomography, when I was at UCLA. UCLA bought a University of Pennsylvania faculty member and his machine and was doing research there. PET scans are not high resolution, but they can be tuned to look at specific neurotransmitters. You can use them, for example, for diagnosing Parkinson's. So they're popular. There may be one in Scranton now. When I came here, I know there weren't, but I think there may be one now. Um, Now an MRI would give you a picture way better than that. Uh, that looks like something from an early Mario game. Um, but that's because it's a PET scan. So it's colorized, and red is usually hotter. So that looks like, I don't know what they're looking at there. Uh, but it's low resolution, but it's pretty magical. So there's discussion of PET scans. You have to use radioactive stuff to do a PET scan, and it has to be produced locally. You have to have a cyclotron nearby, and you run to the PET scanner where you are, and they inject it in you, and it's a deal. Now, fMRIs, um, functional magnetic resonance imaging, they look at blood flow. Now, it says MRI, and MRI is high resolution, certainly compared to a CAT scan, which isn't discussed in here. But fMRIs are not high resolution. They're a bit more like the PET scans. But as you make an fMRI, you take an MRI. So they usually overlay the relatively grosser data from the fMRI on top of a beautiful MRI. So that's most of what you're looking at there is the MRI, and it's beautifully detailed. But the red areas are less detailed, and that's the functional. Oh boy, yeah, read this. This is uh, relevant. So that looks something related to what was at the beginning of the book. That's an fMRI. A friend in the psych department used to have a picture outside his office of research they did with dead fish, and they did fMRIs in dead fish and found parts of the brain that were more active in dead fish. They're dead, Jim. They're not more active. That's an artifact. So you have to be careful with this. fMRIs have definitely been over interpreted. Definitely. Oh, and we're done. Homeostasis. Okay, it talks about neurotransmitters acting a brief period of time because they go small distances. Neurons also release chemicals that can act more like hormones and can go longer distances. But we're focusing on the neurotransmitters here. And there's receptors and dividing the nervous system. And there's some famous neurotransmitters that are important for the course, nigrostriatal, Parkinson's, and boy, that video, L-DOPA. It's too bad that that miracle drug doesn't fix things. Mesolimbic in terms of reward, GABA, alcohol, story of neurotransmitters. So, this was a very thin chapter in terms of content, but I think it was very nicely done. I hope you enjoyed it, and I will now take several hours to save this.